Thanks for giving us the opportunity to present our work. I will talk about our background and the aim of the study, material and methods, results, finally conclusion, and our future aim based on this study. Our background summarized as follows. Histopathological evaluation of morphological features of breast cancer play a central role in the diagnosis of the tumor. Assessment using digital image of histological type, for instance, invasive ductal carcinoma, invasive lobular carcinoma, and histological grade. We used for a grade Nottingham grading system as it is the most common and it is approved by World Health Organization, are a strong prognostic factor in patients and guide clinical management. There was a concern about a grading system as a subjective nature of the technique, as there was a variation among the pathologists to be consistent in grading. So, combination between genotypic description of assessing morphological feature with some robust analytical approach could improve grade prognostic performance and also make it more objective and increase its reproducibility. For more illustration of Nottingham grading system, as it is mentioned in some details in the previous talk, I will go through it in brief. Nottingham grading system is a summary score of three main components: nuclear polymorphism, tubule formation, and mitotic count. Each component were assessed into three scores: score 1 at the low, score 2, and score 3. These three images is a part of digitally scanned breast cancer cases and assessed into three different score of tubule formation. This is a score 1 as it's seen high tubule formation, and score 3 low tubule formation, score 2 between 1 and 3. Second, nuclear polymorphism. There was a marked variation. There was a less variation in score 1. 3 showed the marked variation, and score 2 in intermediates. Finally, mitotic count, which assess after reading 10 high power field, depending on count the mitotic cell, also assessed into three scores: score 1 at the low, score 2 intermediate, and score 3 high. This study mainly based on whole slide image. As we all know, visual microscope using digital whole slide image technology through which glass slide of histological specimen are digitally scanned at high resolution for viewing on a computer screen. This is widely used in research today and has a lot of advantage than traditional light microscope, high quality annotation tools, multiple slide of the same case. Also virtual education, also building model as it is used in this study. Our aim defines specific gene that can predict the grade and its component using whole slide image and messenger RNA sequence data. Before I talk about our material and method used in this study, I will give brief information about data set that using in this study. TCGA data used as the training set, the Cancer Genome Atlas, and the Metabric data molecular taxonomy of breast cancer international consortium, and used as a validation set. TCGA data include nearly 800 breast cancer cases, while Metabric data include 2,000 breast tumor, included 300 cases of Nottingham cases. Both have well-characterized molecular transcriptomic and proteomic data, which publicly available for research community to use. Our material and method: number of cases, 743 cancer from TCGA has both RNA sequence data and whole slide image. Whole slide image, also whole slide image were downloaded and scanned and grading by specialized breast pathologist, one of our group. Grade was used as a target variable with over 20,000 messenger nucleotides and candidate predictors. And information theory was used to select the top genes that highly associated with grade and its component. This information theory has the advantage of limiting the number of genes selected. It's chosen the gene depending on the highly ranked or most significant. RNA-based model was trained on whole slide image in the TCGA data, and the grade was predicted in the Metabric data. Our results: This dendrogram based on cluster analysis showed a set of genes that highly associated with grade and its component and included for a comparison Ki67 and estrogen alpha. As it is known, both of these genes highly associated with tumor morphology and its aggressiveness. From these genes, 3 genes can predict L-grade, centromere protein A, which it is required for targeting the centromere that specify the mitotic behavior of the chromosome. Second one is the TTC8, which play a central role in cell differentiation. Finally, GTP binding protein, which play a role in cell signaling. These results were confirmed from the TCGA data, as both 3 genes showed highly significant association with tumor grade, high expression of centromere protein A, highly associated with high grade. Also the same result for GTP binding protein, while TTC8, high expression, highly associated with low grade. We obtained the same result in the metabolic data as a confirmation, as it is seen in this table. In metabolic data, this row curve showed the high specificity of this model to predict the grade, whether it is high grade or low grade, which assessed using digital image, as the area under the curve equal to 0.8. This box plot showed the expression of 3 genes as a cluster highly associated with high grade. Sorry for that. Regarding the grade, using whole slide image or digital image could improve outcome prediction. This Kaplan-Meier survival curve showed the high expression of 3 genes significantly associated with poor prognosis. The second part of our results, the grade component, these 3 genes showed highly significant association with nuclear polymorphism, aurora kinase A, which found at the centrosome and spindle poles, calcitonin 2, which related to calcium cell signaling pathway, and finally BHF21B, which it is interacted with metal ion. These results were obtained in the TCGA data, as it is shown. High expression of aurora kinase A highly associated with high score of nuclear polymorphism, while calcitonin 8 and BHF21B high expression highly associated with low and intermediate score. Also, when we made the same analysis in the metabolic data, we obtained the same results. This box plot showing the high expression of these 3 genes as a cluster, highly associated with high grade. The second component of grade is a mitosis. These 3 genes showed highly significant association with TTTK, which associated with cell proliferation, FAM161B, which maintain the cytoskeleton of the cell. Finally, CyBird-D1, which it is iron regulating protein. As it is known, iron is highly required for all cell divisions. We obtained this result from the TCGA data. As the TTK gene showed a high expression highly associated with high score of mitosis, while FAM161B and CyBird-D1 high expression highly associated with low score of mitosis. The same result in the metabolic data. In metabolic data, this box plot showed the expression of these 3 genes as a cluster highly associated with high grade. Final component of grade is a tubule formation, and these 3 genes, MC, M5, which participate in cell cycle regulation. The second, C9ORF89, regulate apoptosis, and finally, CDH1, which it is control the cell maturation and movement. In TCGA data, we obtained this result that MC-M5, high expression highly associated with highly associated with high score of tubule formation, and also for cadherin 1, while high expression of C9ORF89, high expression highly associated with low score. In metabric data also we obtain the same results. Finally, in conclusion, based on digital pathology, a model was built and then validated using another data set completely different or completely independent from the training set. The grade and its component was a target. So whole slide image were used to train 3 gene signatures: centromere protein A, TTC8, and GTP binding protein. In metabric data, these genes showed significant association with patient outcome. A digital image shown the ability to transmit sufficient information which leads to objective prediction of patient outcome. A combination between using virtual microscope and the molecular profile could be adjuvant and increase artificial intelligence performance in the digital pathology. Finally, our future aim based on this study, more integration between assessment of morphological features using digital image and the molecular data. Hope it will have more objective role and enhance our understanding of breast cancer biology, refine breast cancer classification, and also improve clinical management and personalized therapy. Thank you, Nottingham Breast Cancer Group.
Thank you. Any question? Okay, I'll ask you one question. You found in this, you mentioned that aurora kinase is associated with polymorphism, which was my first question. So this is aurora kinase is one of the proliferation marker. So it was associated with polymorphism as well in your study, isn't it?
Yes, highly associated with high score of polymorphism.
That's good. Okay. And did you find the Ki67? Was it one of the marker? Ki67.
We use the Ki67 as to be a comparison, to make a comparison when we select these genes.
Okay.